Good day. It's my great pleasure to be with you here today. I'd like to thank the organizers for inviting me. And what I hope to do, even though it's remote, is I hope to share with you the HIV pandemic and the COVID-19 pandemic when these two pandemics collide. And as we think about the collisions of these pandemics, I'm going to share with you some aspects of those collisions at the level of health services, at the clinical level, as looking at how clinical outcomes from COVID-19 are impacted by HIV. And then I'll talk about some of the immunology and the virology and looking at how that is impacting prevention. And I'll end off with just touching on three key lessons from HIV for COVID-19. So let's look at the global situation, just as a scene setter. In HIV, we have about 38 million people living with HIV globally. We have, in the course of last year, about close to 700,000 deaths and about 1.5 million new infections. Compare that to the COVID-19, where since the start of the pandemic in January of last year, we've had about 179 million cases with about 3.8 million deaths. So we certainly are looking at two serious major pandemics in their individual capacities. So what happens when they collide? Well, let's look at the impact on health services. Well, the first thing is that what we have seen is that those countries that have developed HIV programs that are focusing their efforts on dealing with prevention and treatment of HIV, they were better placed to some extent in responding to COVID-19. And let's just look at some of that capacity that was able to pivot from HIV to COVID-19. Let's start with diagnostic platforms. The same PCR diagnostic platforms with the Roche and Abbott technologies that were used for viral load are also being used to diagnose COVID-19. The whole genome sequencing capacity that we've set in place in order to look at drug resistance for antiretrovirals is now being used to do phylogenetic surveillance for SARS-CoV-2. And if we look at the vaccine clinical trial infrastructure that we have built over the years, that clinical trial infrastructure has pivoted and has been the driving force for many of the COVID-19 vaccine trials across the world. So here are just three examples of how HIV and the preparations in the HIV response were able to assist countries to better respond to COVID-19. Now let's look at what has happened in the COVID-19 response that has impacted HIV services. And I'll draw on some of our own data that we've published in Science and in The Lancet. So let's look at, just to start with, the impact of lockdowns. So lockdowns, especially when we have stay-at-home orders, they have significant impact on HIV services. Not because there's a particular challenge that the hospitals or clinics are closed, indeed they're still open, but there's a high level of hesitancy. People are hesitant to go to health services because that's where the COVID-19 patients are. So that hesitancy has translated into impacting negatively on HIV services. And so if you just take in a survey and a study we undertook in 65 health services in South Africa, HIV testing declined by about just over 47%, and antiretroviral treatment initiations similarly dropped by around 46%. Fortunately, we didn't see any marked changes in antiretroviral treatment collections, and patients tended to become somewhat innovative in figuring out ways of ensuring that they had continuity of medical supplies. However, in some other parts of South Africa, there have been anecdotal reports that treatment services were interrupted and access to antiretroviral treatment was interrupted during the most severe periods of the lockdown. So we've looked at the collisions at the level of health services. Let's look at the collisions at a clinical level, and let's look at how HIV impacts COVID-19 clinical outcomes. So in the Lancet, there was this overview that was published that just put together all of the various studies. Most of them are quite small studies, and I'm not going to go through them, but I've provided you with the reference so that should you ever wish to look at that data, you can do so. But what we saw is that most studies showed that people living with HIV were not really impacted in terms of their COVID-19 outcomes. However, those results, that lack of impact was largely in the smaller studies. In large studies, what we tended to see is differences in clinical outcomes appearing. So it seems that whatever the impact of COVID-19, whatever the impact of HIV on COVID-19 outcomes is, it's probably quite a small impact and is evident only in the larger studies. So let's look at one of those larger studies. This is a study looking at the situation in the UK. In a retrospective cohort of around just over 17 million individuals, of whom about 27,000 had HIV infection. What was seen is that people living with HIV tended to be more often male, more often black, more often living in poorer settings. And of the deaths that occurred, there were about 25 that occurred among HIV positive patients. And when looking at this analysis, that COVID-19 deaths were increased by about 2.9-fold in patients who have HIV. The challenge is that that difference is confounded. It's confounded by race, it's confounded by gender, it's confounded by socioeconomic status. For example, if one just looks at mortality among the black population, it is 4.3. Compared to amongst those who are not black, it's 1.8. So there seems to be an increased mortality in patients who have HIV. And that difference is somewhat confounded by a range of factors. A large study in South Africa shows that the difference between mortality in HIV positive individuals and HIV negative individuals is about twofold. But once again, in this study, which is a pretty good study, large study done by Marianne Davies and her colleagues in the Western Cape, that study is also confounded because many of the patients who had HIV had other comorbidities. And it's not easy to just tease out the multiple comorbidities to look at the actual contribution of HIV. But even after adjustment, remember that HIV is largely in the younger population, and the COVID-19 deaths are larger in the older population. So that age difference means that HIV, even if there is an increased risk, is still only contributing a very small proportion. And that's what was seen in the study in Cape Town, that only about 8% of COVID-19 deaths could be attributable to HIV. So it seems that there is some association, probably a small association, and those associations are quite difficult to tease out in the midst of multiple confounders. Let's now look at the third level of the collisions, at the level of immunology, at the level of virology, and look at how all of those are impacting prevention. Let's start by looking at natural and vaccine-induced immunity, and look at in particular how in HIV-positive individuals who are immunocompromised, what is the impact on natural immunity? Well, what we have seen is that people with HIV have tended to have lower neutralizing antibodies against SARS-CoV-2. The extent to that difference seems to be small, but it's certainly there and is one that may impact in terms of the risk of reinfection, but that needs to be empirically demonstrated. Of greater concern is that individuals who are severely immunocompromised as a result of HIV, let's take this patient here in a case report published in Lancet HIV, that in a patient with uncontrolled HIV, with a high viral load in the region of just over 800,000, with a CD4 count as low as 20, this individual was given 2 doses of the Pfizer vaccine. But after 2 doses in 28 days, still no reactivity. We saw no IgG reactivity, no SARS-CoV-2 neutralizing activity, and no ELISpots. So there was no B-cell response and no T-cell response. And so that becomes quite an important issue, that individuals who are severely immunocompromised from HIV are not responding to vaccines. Let's look at one particular immunocompromised patient, and this is from a study in South Africa. In this particular individual, who was quite severely immunocompromised, CD4s of only 6, in this particular patient, there was persistent SARS-CoV-2 infection. It was persistent for over 7 months, and during all of this time, SARS-CoV-2 could be cultured from the swabs from her throat. So looking at this particular patient with a CD4 count of 6 and a viral load of around 34,000 and a SARS-CoV-2 CT value of 18.5. 18.5 translates into a pretty high SARS-CoV-2 viral load. What we saw is that at the end of those 7 months, when she was put onto effective antiretroviral therapy, switching from her regimen to a dolutegravir-containing regimen, we saw her viral load decline. And with the decline in viral load, she also cleared her SARS-CoV-2. If you look at that situation, in that 7 months, we saw repeated mutations occur. So let's look at those mutations. In this particular individual, she was initially infected with a D614G variant. That's the variant that's occurring all over the world. Over the course of those 7 months, there were 11 mutations in this individual. In particular, I want to focus on 3 mutations because those 3 mutations are really important because they constitute the beta variant. E484K occurred in this individual very early on. On day 6, she already developed this mutation. That's a key mutation because it enhances immune escape. She developed the N417 mutation at day 71 and the 501 mutation at day 190. So this patient, over 7 months, with all of the mutations, recreated the beta variant. And so it just indicates to us the central role of immunocompromised individuals in generating new variants, and how in this particular individual we're seeing how the beta variant is actually being created, at least in the RBD mutations. It's interesting that this patient during those entire 7 months was on antiretroviral treatment but had poor adherence. In fact, over those 7 months, at only 2 time points were her antiretrovirals even detected. And she had multiple mutations that conferred antiretroviral drug resistance. She had mutations against tenofovir, FTC, and efavirenz. So let's end off with touching on 3 key lessons that HIV has for the COVID-19 response. So the first I'll touch on is about know your epidemic, know your response. Now, we have spent decades refining this, fine-tuning it so that we really know about how to tackle our HIV epidemic. So I've shown you here on in the map that you see the HIV hotspots in northern KwaZulu-Natal and how we use those hotspots to target our HIV interventions. And we have whole programs and guidelines about how to use surveillance, how to use data to guide our HIV response. If we look at SARS-CoV-2, we're doing exactly the same in South Africa. In this map, you're looking at Khayelitsha, just outside of Cape Town. And what we've done here, the Western Cape government has individually mapped every case of SARS-CoV-2. And what you're seeing is a hotspot. And when you look at these hotspots in Khayelitsha, what you see is that the supermarket in Khayelitsha is a major source of transmission, and it became a point of intervention to slow down the spread of SARS-CoV-2 in this area. So, know your epidemic. Know your epidemic at a granular level and refine your response to target it appropriately. The second is we've learned about the importance of the centrality of the scientific response and the use of scientific evidence in the response. In HIV, we collate enormous amounts of data, we use it to define our objectives, we draw detailed plans, and so we've used data to define our response. In SARS-CoV-2, we've done similarly. In South Africa, we've used data to define the 8 stages of our response. And we've known, and we've seen in HIV, how when politicians prevaricate, when they go into denial, the impact that that has on HIV. And we saw that in South Africa in the past regime. We saw that similarly for SARS-CoV-2 in the US, where we had the promotion of this notion that, oh, the virus is going to disappear, or let's use hydroxychloroquine. So when politicians go into denial and when they prevaricate and promote non-scientific methods and non-scientific thinking, it impacts on our epidemic. And so the scientific response is critical. The third that we've learned from HIV, that is a particularly important lesson for SARS-CoV-2 and the COVID-19 response, is the importance of mutual interdependence. And we've seen that in HIV. And if you just look at, and I'll quote you from the UNAIDS report, that the AIDS movement demonstrates that with a shared vision, shared responsibility, and through global solidarity and leadership of people living with HIV, affected communities, and individual action, we can change the course of history. And we've done that. In HIV, we've shown what global solidarity can do. We see it in the Global Fund, in UNITAID, in PEPFAR, how wealthy countries are contributing funds so that poor countries can gain access to treatment and save lives. We're using that as a template. The WHO, Gavi, and CEPI created COVAX to create a global mechanism for vaccine equity. The challenges we are experiencing now is that we are not seeing that global solidarity. Rich countries are not sharing and not providing and supporting COVAX to the extent that's needed. So let me throw down this challenge and quote from Pope Francis. And this is from an op-ed that he published in the New York Times. And I'll quote him. "The pandemic has exposed the paradox that while we are more connected, we are also more divided. To come out of this crisis better, we have to recover the knowledge that as a people, we have a shared destination. The pandemic has reminded us that no one is saved alone. What ties us to one another is what we commonly called solidarity. Solidarity is more than acts of generosity, important as they are. It is the call to embrace the reality that we are bound by the bonds of reciprocity. On this solid foundation, we can build a better, different human future." Let me reiterate that point, the bonds of reciprocity. What I do impacts your risk. What you do impacts my risk of COVID-19. We are fundamentally interdependent. One country cannot solve the global pandemic of COVID-19. We have to do so as a global effort. What one country does impacts another country. So we stand together, and it's only through standing together, and we've learned that from HIV, that we can solve the challenges of COVID-19. Thank you very much.